Hi everyone, my name is Molly Taylor and I'm a senior biology major here at Western Kentucky University. And today I am pleased to share with you my undergraduate research entitled The Effect of Age on Neurological Inflammatory Responses to Sleep Fragmentation in Mice. First, I want to talk about my clinical correlate or my real world application to my research, and that is obstructive sleep apnea. Obstructive sleep apnea is defined as the intermittent airflow blockage during sleep. And as you can see in the bottom of that picture there, a person with obstructive sleep apnea has a blocked airway while they sleep. And this can cause intermittent hypoxia, perturbations in sleep continuity, and can ultimately lead to cardiovascular disease and neurocognitive decline. Some of the major risk factors for OSA include increased age and obesity. Inflammation and sleep apnea. This is what my research is specifically about. So, sleep apnea is categorized as a chronic inflammatory disease, and this means that people that have obstructive sleep apnea are going to have higher chronic levels of inflammation or swelling in the body compared to those that do not have OSA. This is so dangerous because underlying inflammation can induce organ dysfunction in patients with OSA, which can ultimately lead to death. And this is one of the leading reasons for mortality in patients with obstructive sleep apnea. Now, TNF alpha is a pro inflammatory cytokine that is used in my research. And cytokines are little molecules in our bodies that are secreted by immune cells that basically regulate our immune responses. There are two major types of cytokines: pro inflammatory, which induce an inflammatory response, and anti inflammatory, which take away an inflammatory response. In the case of TNF alpha, it is a pro inflammatory cytokine that has implications in sleep regulation and is typically enhanced following sleep deprivation, making it the perfect model for my research. The purpose of my study was to assess whether age affected neurological inflammatory responses to sleep fragmentation in mice. And my hypothesis was that neuroinflammation would be exacerbated in older mice compared with young mice in response to sleep fragmentation. Materials and methods. So I used 36 male C57BL/6J mice. This line of mice is typically used as a model for human disease studies. So I had four groups of mice. I had a young control group, a young sleep fragmented group, an old control group, and an old sleep fragmented group. There were 9 mice in each group. My experimental setup entailed a 24-hour study, and during that 24 hours, all of the mice were put in their respective groups in sleep fragmentation chambers. These sleep fragmentation chambers were equipped with a computerized swipe bar, and in the experimental condition, these experimental swipe bars were turned on, and they were programmed to swipe across the cage, across the bottom of it, every 60 seconds. And this induced the sleep fragmentation by causing the mice to have to move around the cage and wake up every 60 seconds. This is what one of those cages looked like with the computerized swipe bar, and that is what one of the mice would look like. Next was tissue collection and RNA isolation. So immediately following the 24-hour study, all of the mice were euthanized with isoflurane gas and then decapitated, and 3 major brain regions were collected, including the prefrontal cortex, the hippocampus, and the hypothalamus. Next was RNA isolation and cDNA synthesis. So we used an RNase mini kit for the RNA isolation and followed that protocol. And then after the RNA was isolated, we checked the quality with the NanoDrop 2000 provided in the Biotechnology Center at Western Kentucky University. After that was finished, we diluted all the usable RNA samples into the same concentration, and then we reverse transcribed them into cDNA using a reverse transcription kit in the lab. After our cDNA was synthesized, we used real-time PCR. We used an Applied Biosystems 7300 machine, which was provided in the Biotechnology Center. And this is what allowed us to look at the relative gene expression of TNF alpha. Our control gene was 18S, and it's a good endogenous control because the levels of 18S in all the cells of the body are pretty much the same. And then our test gene was TNF alpha, of course. This is an example of a 96-well plate that was used in the real-time PCR machine. So I loaded our cDNA, our genes, and a master mix into these wells, and then I put the plate into the real-time PCR machine according to protocol, and it ran a sequence and allowed amplification of the genes, and quantification of the genes. So my statistical analysis, I did my relative gene expression. So after the real-time PCR was performed, it gave me several different values, and I did what was called a comparative CT analysis, and I took the delta-delta CT of the ratios between the 18S and TNF-alpha levels. And that gave me the graphs that you will see in the results section. Then I used a two-way ANOVA test. So a two-way ANOVA test is a statistical analysis that showed the significance of all of the factors of the study. So it had sleep fragmentation alone, and then age alone, and then it looked at sleep fragmentation and age together. And it basically told me whether my results were significant or not. And for results to be significant, the p-value had to be less than 0.05. Now let's look at the results. This is the prefrontal cortex, and as you can see, the sleep fragmented condition, TNF-alpha levels were higher than those with no sleep fragmentation. But according to my two-way ANOVA test, sleep treatment was significant with a p-value of 0.0396. But as you can see with the graph, age and treatment with age did not have a significant effect on TNF-alpha expression in the prefrontal cortex. In the hippocampus, you can see again, there were higher levels of TNF-alpha expression in the sleep fragmented group. But according to the two-way ANOVA test, as you can see, there were no significant p-values in the hippocampus. So that meant that TNF-alpha expression was not significant in the hippocampus. In the hypothalamus, you can see that, again, the TNF-alpha expression was higher in the sleep fragmented group than in the non-sleep fragmented group. And according to the two-way ANOVA, the sleep treatment and age separately both had a significant effect on the TNF-alpha expression, but treatment and age together did not have a significant effect, according to the p-values. Here are my conclusions. So in the prefrontal cortex, like I mentioned, there was a significant effect of sleep treatment on TNF-alpha gene expression. No significant effects on age or age with sleep treatment. In the hippocampus, there were no significant effects of TNF-alpha expression. And in the hypothalamus, sleep treatment alone and age alone both had significant effects on TNF-alpha gene expression, but there was no significant effect on age with sleep treatment on TNF-alpha gene expression. This partially disproves my hypothesis in that age is not associated with an effect on TNF-alpha expression in mice, in sleep fragmented mice, but it does, it is significant, and it is continuous with all of the information that is out there with inflammation being higher in the sleep fragmented mice than in the non-sleep fragmented mice. So some improvements to this study. Larger sample sizes are always better for studies in most cases, and having more mice to compare it to, it just gives you a larger set of data. And then analyzing the differences in males and females. I think that this would be important because males and females respond differently to inflammation in so many ways. And then also doing a longitudinal study. My study was more acute as it was only 24 hours. And as sleep fragmentation and sleep apnea are chronic diseases, a longitudinal study would be good to have a more accurate representation of that data. Some future implications, we can have a better understanding of the inflammation response in sleep apnea and in sleep fragmentation. We can use this to implement better treatment for OSA patients and drug optimization as well. And we can have a better understanding of obstructive sleep apnea disease progression. Here are my acknowledgements. I'd like to thank a few people, especially Dr. Noah Ashley. He is my research advisor. And then Dr. Banga and Dr. Day, they are on my thesis committee. And then Miss Naomi Rowland and those in the Biotechnology Center, the WKU Biology Department, the Mahurin Honors College, and the members of the Ashley Lab. Thank you so much for listening to my talk, and I hope my research interested you, and I hope you guys have a great rest of the conference.